we'll see how this, I think that's perfectly fine now. So the fateful day has arrived. You had to read something in my book. And we're going to discuss the first three chapters today, material from the first three chapters. But as usual, questions that derive from what we've done before are perfectly allowable and welcomed by me. So if you're thinking about, hopefully the whole purpose of this, or my goal for this, is to integrate the material all the way from day one, where we talked about species concepts, out to the session one, to this eighth session where we will talk about conservation implications of genetic exchange, and which has to do a lot with species concepts and our philosophy of species, and then also our own evolution, and we'll talk about that some more in the last session. But I'm hoping that all of this makes sense integrated together at some level for you. I know that there's a lot of information, huge amount of information, but what I try to do, I'll give you a little bit of a philosophy of why I write books, okay? There are a few reasons. Obviously, communicating a message, conceptual framework is one of them. There's a very selfish reason for me, and it's not money, because you don't make any money off of these books like this. Undergraduate introductory biology texts you can turn into a career, make hundreds of thousands of dollars every year on those, but I don't write those. I don't want to write those. But it is selfish in the sense that it comes a point in time where, for me personally, I want to try to synthesize in my own mind all the data that I've brought in, as much of it as I possibly can. And so look for patterns, look for processes that overlap between different domains of life, those sorts of things. And so that's on that side. But in terms of communicating, the goal for this particular book was to bring us up to date. Really, this is sort of, well, they asked me to write a second edition of my 2006 book, Evolution Through Genetic Exchange. And I said, okay. Oxford came to me and asked me to write that because it had been for that kind of a book, a lot of people bought it. And I told them, okay. And so you write a book proposal, you send it in, it's reviewed by outside reviewers outside of your publisher. They come back and tell you what you need to add or what you need to take out, they feel like. And so you modify your book proposal and then you start writing. In my case, I'm always already writing or thinking about the writing process. And when I started writing, I realized it was going to be a very different book. Even though the format is very similar to my 2006 book, actually, in terms of chapter structure, the kinds of topics and the way I have to approach those now, because of the revolution in how we think of things since 2006, I mean, our evolution, for example, that paradigm has totally shifted. And so, but it's even more than that. It's, eukaryotes having all of these different pieces of genome in them from prokaryotes, which we knew nothing about in 2006. In fact, I kept bugging my bioinformatics. We have an institute for bioinformatics, and I kept asking the director, who's a member of our department, okay, have people found this and you've seen the manuscripts, but I don't know about them? And she kept telling me, no, I'll let you know. I really drove her nuts. And then finally she popped up and said, Mike, there's a Nature paper coming out, and it's actually passed the gradient and the restrictions that we've put on all of our papers for being really, really good. So it's that sort of thing that really was a conceptual shift because of methodological and data analyses and data gathering. And that's what's going on still. And I kid about epigenetics at sometimes with my epigeneticist colleagues, but I do indeed feel like it's going to be a major revolution for us when we start really understanding what's going on there. So This book hopefully will be okay for about 5 years, but I doubt it. It'll be behind the curve probably in another 2, is what I'm guessing, just because of the speed of things that are being discovered. So what I'm thinking is I may write it one more time, this kind of a book, one more time before they put me in the ground, but we'll see. All right, first 3 chapters. And what I want to start with, though, is I mentioned this to you, I want to start, and I'm not going to have a lot of PowerPoint presentations. This is it, and it's short, okay? So I really need you guys to be asking questions from the material, interacting in that way. So we're going to really try to get back into that format. The last 2 sessions, obviously, were designed to be lecture. So what I want to talk about, though, today, start out with you, is the incredibly important subject of how do we test alternative hypotheses in terms of genetic exchange, and what has alternatively or alternately been called incomplete lineage sorting, retention of ancestral polymorphisms, or deep coalescence. You'll see all of these in the literature. This is the most commonly used, but I think it's nonsensical, except for a theoretician who does math, but we're going to stick with it because it's the most commonly used. But it's like linkage disequilibrium. It doesn't mean what it says. And by the way, with linkage disequilibrium, I told Dick Lewontin, who invented the term, that. I think I may have mentioned it to you. And I said to him, I don't understand. Now, this is a guy at Harvard, and I shouldn't have probably challenged him, but he actually signed all my books of his that I bought. So he was actually quite kind about it. He said, I don't know what I was thinking when I named it linkage disequilibrium. I said, I don't know. He said, I agree with you. It's not a good terminology. Same way with this. Incomplete lineage sorting, if you're into the math of coalescence, it just, you see it. But this means, I think, is much more descriptive, and this is what we were saying long before incomplete lineage sorting when we were working on phylogenies back in the 1970s. And so this was replaced with this, I think, because it sounded so much sexier. Okay, but that's okay. In the literature, that means that, and deep coalescence is even worse, but it's something you see. Mostly mathematicians. Okay, so let's talk about, and these are very simple examples. I said that the other day when I drew up, incomplete lineage sorting, how it could affect phylogenies. Okay, I know these are incredibly simple, but when you do 1,000 loci, this is what you're doing, right? If you're looking at 1,000 loci and you're analyzing them and trying to understand what the patterns mean, if there's allele sharing, this is what you're testing for. So, genetic exchange and phylogenetic discordance. So, incomplete lineage sorting and genetic exchange have historically, those 2 alternative hypotheses, have historically been tested with gene genealogies first, and primarily people still do it. Once again, please understand, if there's a paper out there that says, we detected this much incomplete lineage sorting, and they only use BEAST, or a format of it, which I've used. We have a paper in review right now, manuscript in review right now. We use BEAST. It's very useful. But it does not test for anything but incomplete lineage sorting. It only estimates incomplete lineage sorting. So if you want to test these hypotheses, don't be like some of the major labs that publish in Nature, Science, and Evolution, and other things like that, who say, incomplete lineage sorting, contributed this amount, and then use the wrong package. Okay, that won't give you the test of this. If you're not interested in it, if you just want to conclude, oh, well, we know animals don't do that, then that's fine. Well, it's not fine, but you ought to test. All right, so if this is the real phylogeny, okay, for some reason we know that this is a real phylogeny, or a species phylogeny, or we know that we're using one as our litmus test, say morphology, And we want to compare all of our genomics to that. Or we have genomics and we want to compare morphology, whatever. But in this case, this is our true phylogeny. We're working with phage and, whatever. So this is, if we have genetic exchange of locus 1 between A and C, we get the wrong phylogeny. We get discordance, okay? If we don't have it, I know this is simple, but if we don't have that or any other exchanges, we get the correct phylogeny. And so now you're looking at a second locus. Locus 1 gives you a different format, okay, than locus 2. So locus 1 and 2 give you discordant phylogenies. Locus 1 gives you a discordant phylogeny to whatever you're thinking about is the right one, and locus 2 does not. Okay. Now, let's look at ancestral polymorphism or lineage sorting and phylogenetic discordance. So if, I mentioned, depending on what organisms you're looking at, okay, whether you're looking at viruses, prokaryotes, eukaryotes, plants, and animals, there's a level of polymorphism, we could call it, or genetic variation expected per individual, per population, per species, right? You estimate it when you do your genomics. Let's say the polymorphism level is 30%. It may be much more depending on how wide your window is that you're sliding along your genome. But anyway, let's say 30%. Well, let's say locus 1 is part of the 70% that are monomorphic. It could be flipped around, by the way, maybe 70-30, who knows. But the point is, with this kind, there's no genetic exchange. The only allele that can sort is 1. All right? It's monomorphic, or it's homozygous, I mean. Okay. Ancestral polymorphism, now you actually have a polymorphism. It's heterozygous down here, okay, at locus 1. And this sorts in this particular way, just by chance. It's coalescence, all right? And you end up with it like that. Once again, you do not get a discordant phylogeny, even though you've had coalescence. And remember, coalescence is real. Incomplete lineage sorting, particularly in younger groups, is going to be there. Okay? You have to estimate it if you're interested in trying to figure out whether or not your phylogeny makes any sense whatsoever. You do something that we used to call, anyway, estimating homoplazy. And homoplasious loci are like this, okay? They're homoplasious. So in this case, and this is due to incomplete lineage sorting, so in this case you have the polymorphism. You can retain the polymorphism. This does not. This allele sorts here. You still have the polymorphism with one sorting there, and from this you get this kind of sorting. And what you end up with is, quote unquote, the incorrect phylogeny. You end up with discordance again. This being discordant from those other loci that you saw with no genetic exchange, but just various forms of lineage sorting. Whereas here you have this, what is referred to as incomplete lineage sorting. And what we mean by that is that this, if given enough time, should end up having just the alleles that tell you that A and B are together by coalescence. Yes? Yeah, I mean, it could, yes, you're talking about genetic drift,
Yes.
sampling error.
In that, there has to be a bottleneck. So that is the only one allele is there. If there's a large population size, then there will have probably go to plus in there. So why is it not clear that there also has to be a smaller population?
Yeah, I'm just using the simplest illustration, but you're right, this could have 1 and 2. And this could just have 1, not even by genetic drift necessarily, but selection or convergence or whatever else.
Also due to drift, right?
No, not necessarily. I mean, you're losing alleles by chance, but it doesn't have to be by drift, right? It could be by population substructuring, it could be by selection, it could be by drift. Yeah. there's a lot of different processes that could lead to this, but what I want to emphasize is it has nothing to do with genetic exchange of any kind. That's the major point here. And this is theoretically what you expect, once again. And what we observe, once again, particularly in relatively young, there's a N sub E, or a generation, time that if you've had enough generations since here, you should not have a problem with incomplete lineage sorting if it's very ancient. So in other words, if you had something way out here and it was sharing alleles up here, that would be more, depending on the genomic architecture, that might be more indicative of genetic exchange. But yes, all of these could be filtering through bottlenecks, but they also could be filtering through selection, etc. Well, environments change. And so, these are originating, the cladistic events here that are splitting these apart, those are speciation events. The speciation event could be reflecting environmental dependent selection, etc. But keep in mind that, once again, in all of the phylogenetic programs that we run, whatever you choose, they're always going to, well, they give you error bars or standard deviations or whatever. That gives you an idea of how much mess is there, whether it's genetic exchange or not. But that's incorporating parallelisms. I mean, we're assuming parallelisms, convergence due to selection, incomplete lineage sorting, and depending on what group you're working on, if it's birds, they almost never conclude integressive hybridization, for example. But they, ornithologists just historically have been that way, which is interesting because, one of our best examples is Darwin's finches kind of thing. So, but anyway, that's, but that homoplasius sort of nomenclature has been around for decades, and it just reflects noise, is what we say. If all we're interested in is drawing phylogenies, and when I say all, I did this for 6 years and I loved it, okay? Doing phylogenetic systematics in mammals. And I love drawing trees, okay? As odd as that sounds. But what we do now that is better, much better, is to do actually networks. Allow it to be a network. Don't force it into a tree-like structure. And if it goes into a tree-like structure, great, but allow it to be a network, and that allows you to also estimate what people often call migration events, which are integration genetic exchange. That's my recommendation to people who are doing this sort of thing. But a lot of people are just wanting to draw the best phylogeny they can and then map character traits on it, which I like. I understand why they're doing that. Yeah. Why does this not indicate it's genetic exchange? Because I'm telling you it's not. What I'm saying is that there are methodologies for testing. We're about to go into them. What I'm saying is that you're getting discordances that are because of genetic exchange, but you can also get those discordances without any genetic exchange. That's the point. Those are your alternative hypotheses that people are testing, or if they're testing for it. Often they're not. Once again, often all they're trying to do is resolve a phylogeny. So if you look at phylogenetic systematics in general, that's plants and animals in general, quite often, like in molecular phylogenetics and systematics or evolution or whatever it is, and molecular phylogenetics and evolution, yeah. If you look in there are very few instances usually where they're testing for this. They just put it together in a package of this is noise, experimental error. We call it a lot of different things. But anyway, I'm just telling you that's incomplete lineage sorting. We'll talk now about how, and in Chapter 3, once again, other ways that people are doing, some of the ways that people have used over the years to test these alternative hypotheses. Yeah?
Michael, this has nothing to do had diverged a long time back, it would still, this pattern is not going to change. The way the alleles sorted into different lineages was not going to change. So why is it that they say that, when lineages diverged recently, like more likely to find incomplete lineage sorting?
Yeah, so what they're actually reflecting is that if you do the math, and the theory is correct in this case, if you do the math and have fewer generations, this is going to be amplified because that inherent polymorphism that's going to be there at, say, 30% of your loci, just through the coalescent process. Now, please understand, they're assuming neutrality. So this is why I'm going back to your question. They're assuming neutrality. And so just through neutral processes of lineage sorting, you're going to end up, the Madisons do a really good job of this, but you have to imagine that you have a swirl of alleles here. And those swirl of alleles, lots of different sub-branches within each one of those just stick figures branches. And within that, the math tells you that as it goes up, it's going to lose allelic composition and richness. But it takes recombination, it takes meiosis, it takes all of these processes. That's why you're losing it. Genetic drift, they really don't invoke selection much. I put the selection in there because I think that convergences, that homoplasic part of that is parallelisms and convergence. So we ought to think that selection is going on. But all this is that from a time point, and this is from the math, not from observation, I suppose, to some degree. From this time point, the expectation with the assumptions of the incomplete lineage coalescence model is that you're going to lose that kind of signal overall. That overall, as you move up a lineage, it's going to only have alleles. The alleles are going to sort out, once again, the math. The alleles are going to sort out and move through that lineage, and you're going to end up with resolved phylogenies. It never happens like that. But because if you're young, there's always noise, there's always incomplete lineage sorting effects, just like you said. But the point is that as you move up, the idea is you get new mutations, they identify those lineages, and overwhelmingly your signal becomes one of the real tree, quote unquote. So, but that's why the problem is amplified and thought to be worse in like cichlid fishes in 15,000 years of divergence, rather than cichlid fishes in a 10 million.
So just to clarify the terminology, when you say retention of ancestral polymorphism, in a situation like this, would it be right to say that the polymorphism is not retained in the resulting lineages?
Yeah, no, it's...
So the zygosity that you started with has disappeared now in the... So would it be right to say that the polymorphism is not retained?
No, it's because the retention of ancestral polymorphisms is a good question. The retention of ancestral polymorphisms is just like incomplete lineage sorting. Incomplete lineage sorting means that you have the wrong polymorphisms distributed because you know the real phylogeny. You have the wrong polymorphisms on the wrong lineages, i.e. starting material polymorphisms. I understand what you're saying. This is homozygous here, this is homozygous. These could still be heterozygous. But once again, for simplicity, I let them be homozygous. But no, this is the retention of ancestral polymorphism term is supposed to say, look, you started out with polymorphism down here, and you end up with those same alleles up here at your tips. That's why people invented the language that way. So, but yeah, I know what you're saying, but we haven't lost any of the allelic variation down here. That's why it was called that. Mm-hmm. You can. This is one of the reasons, actually. And this is a good aside. I mean, this is a good thing for us to discuss in terms of methodology. This is why phylogenetic systematists will always argue now that you really have to sample well. And I know, I mean, it's only money, but it, you know, I mean, it's like, really? But that is one of, your point is excellent, because that is one of the reasons they argue for every branch in this, you really need to try to do the best geographic sampling, even if it's only one individual per population, but across a geography, multiple individuals per population, etc. Makes it difficult, but that's, they say that without that, what you just mentioned could be a real problem in their phylogenies. So you have to balance it, okay? How much money do I mean, seriously, it's how much money do I have, how many lineages can I do, and get a, you know, and what will my power be in estimating my phylogeny when I come out of X, Y, or Z analysis. You can simulate that ahead of time, but it's true, yeah. Other questions before I move on? These are great. Okay. So, we want to try to discuss, or sorry, we want to discuss now, or I want to discuss now, and try to decide how some of the ways that people use this comparative approach, where you use both phylogenetic discordance, at least starting with this one, phylogenetic discordance and something else. Because phylogenetic discordance does not tell us on its own, remember, that we're looking at introgressive hybridization in plants and animals, okay? Right? So once again, I'll harp on this. You can read in Chapter 3 about a lot of different kinds of methodologies that people have used. Some of them very recent. I mentioned I reviewed a paper that just showed up in PNAS not long ago, where they were using another kind of methodology. And so these are changing all the time. So if you ask me what the best way to do your data, I told you this, that I got this asked a lot in China in the 4 courses. I will tell you the same thing I told the Chinese folks, and that was, I don't know. You need to go to the literature. I can tell you general ways to do it, but I don't know what the newest one is going to be when you're analyzing your data. There are stock ones, once again, but remember, look at the assumptions and what the methodologies do. Make sure they're doing what you want them to do, the packages, okay? All right, phylogenetic discordance in ducks. So in this case, what we have are trees, morphology, basically taxonomy, okay? Morphology for these kinds of ducks, okay? And mitochondrial DNA from those same ducks, clades of ducks. Hopefully you can see these dashed lines. Those indicate discordances between regions of phylogenetic discordance between those trees. Okay, based on very different data sets. So, of course, these could be lineage sorting or introgressive hybridization, right? Could be causing this. So what else do we know? Well, first of all, wild duck species are noted for hybridizing. More importantly, which I don't have up here, is that these ducks hybridize amongst themselves contemporaneously. Okay, ducks actually, very high proportion of duck species. It's the highest the grants noted. I've forgotten 60% of them or something like that of all duck species introgressively hybridize with somebody else right now. Okay, sort of like canids, although dogs are worse. Every dog's hybridizing with something else. So with these additional data and the phylogenetic discordance, we would suggest then that it's a portion of this, and then you would need to go in locus by locus, except it's just mitochondrial DNA, but look at the geographic, and we'll talk about why I say that, geographic regions of these, etc., and it suggests that hypothesis of extensive introgression in here, and they've tested it now with many nuclear loci, etc., and indeed they have introgressively hybridized as they evolved. So it's divergence with genetic exchange with the ducks. Okay.
Actually, in this case, both morphology or phylogeny using mitochondrial DNA can give you an erroneous phylogeny. Right? With morphology, you're always bound to run into errors because of convergence. And even with mitochondrial DNA, if you to begin with, if you don't know if it's because of incomplete lineage sorting or because of genetic exchange, you're going to end up with a different phylogeny. So comparing these two phylogenies itself, do you think is the right approach to conclude, hybridization?
Yeah. So the question is comparing mitochondrial DNA and morphology, but I would extend it out even further in terms of comparing any two phylogenies based on very different data sets. The short answer is phylogenetic discordance actually is incredibly powerful. It tells you something is going on there. And yes, morphology could be affected by selection, as could mitochondrial DNA actually. And mitochondrial DNA could be affected more by not necessarily incomplete lineage sorting, but the fact that its N sub E is so low, right? It's 1/4 because of its maternal inheritance in animals. And so there's a number of problems, quote unquote, with any data set you'll construct, that you'll put together. Nuclear DNA, it's often because of recombination. Nuclear loci are pains in the butt because of recombination and breaking up what's the phase and all this sort of thing. Any data set you use will have those, some noise, if you will, in the data set that you have to try to accommodate within your phylogenies. So do I think this is appropriate? Absolutely. I think it would be even more appropriate, even better, when they then compared the nuclear DNA, but that doesn't, and what they found, lo and behold, it's the same kind of patterns. But what I would say is that what we don't know with just this is whether, and knowing that they're hybridizing, what we don't know from this and this is selection for the morphological traits. There's going to be male plumage. Is that included here? I don't remember. And that sort of thing, their trait selection, they're trying, well, of course, when I grew up in phylogenetic systematics, we were always harping on trying to use neutral characters, right? We just wanted them to sort out with that was, of course, we knew we weren't, especially when we were doing morphology, which we were at the time too. So yeah, I think this is really powerful to tell you something's there, and then you go into some other ways of doing this. And I will tell you, discordance among gene genealogies is the starting point for most tests for this. It just is.
Sorry, I just have one more question.
Yeah.
How different do the three topologies need to be for you to call it a discordant phylogeny? In this case, well, you can say that it does seem very different. The three topologies are resulting from two different methods. But in some cases, you might have very minor differences. I mean, there would be a whole gradation, right? So
Yeah.
Does it just come with experience, or are there some rules of thumb?
No. There are methodologies for estimating whether or not the topologies. Well, first of all, you get an estimate. In most of the analyses, you get an estimate for the number of trees that are produced that are equal length or have the same probability or whatever, and so you get 10,000 probably, okay, or however many, unless you constrain it, and then you can drop that number, but is that reality? But there are a lot of trees out there that are just slightly different from one another, but they have the same probability, same length, whatever it is. Those are discordant, but they fall within the same tree. They fall within your best answer. So in that case, you have one data set that's like that, right? That's just one set of whatever is giving you that many different phylogenies. And so you represent that with error bars, whatever you want to call it, whatever you call it. You represent that. But what you then can do is if you have another data set and you look at it, you can actually test there's methodologies for testing, is this topology significantly different in some ways? Significantly different now. I mean, there are a lot of Bayesian analyses too. But yeah, it's a very good question. But we did it by eye. this lineage before we had those kinds of analyses. Yeah.
Would pattern based on mitochondrial DNA be more resistant to select discordance due to selection and due to parental effects?
Well, here's the deal. We have a dogma, another dogma, and actually John Avis talked about this years ago when he was talking about phylogeography. But the dogma is there's a lot of dogmas about mitochondrial DNA. It's always maternally inherited in animals. You know, it's neutral. It's this. It's that. And the likelihood of mitochondrial DNA overall as a unit being neutral is 0 because it's packed with genes, right? So in real terms, mitochondrial DNA, and in experimental situations, we know that you can end up with cytonuclear problems even within the same species if you have different haplotypes of mitochondria. So it's not neutral. And so that's one thing we need to keep in mind because but it's a dogma that's really stuck. And so people, because we're talking about zoologists again, so when we saw our mitochondrial DNA go to introgress and go to fixation on the other side of that, in that other species, we concluded that it was not likely to be selection, but rather just bottlenecks. It's 1/4 N sub E, you know, it's just going to fixation by chance. It might have gone extinct and not shown up. But it really doesn't fit the pattern when we look at those grasshoppers. It doesn't fit the pattern that we see in hares and that have this really high frequency. All to say, we still have to be concerned about selection and convergence due to selection in mitochondrial DNA, I would argue anyway. What mitochondrial DNA does give you, because of the 1/4 N sub E, is a prediction that if you have incomplete lineage sorting, it's going to be less of a problem in mitochondrial DNA the further you get away from the divergence time. There'll be faster coalescence in mitochondrial DNA than in nuclear alleles, on average. So anyway, no, it's so much easier to simplify our models, and we have to do some analyses. And one of the simplifying assumptions for these kinds of analyses or population genomics using mitochondrial DNA is that it's neutral. And how can it be neutral? I mean, I asked people this years ago. You know, I'm always asking bad questions, but I asked, how can it be neutral? There's nothing there but genes. I mean, it blows my mind, you know, to look at it, but I don't know. I mean, I understand the point that maybe it can act like a neutral molecule because when it changes, whatever those genes are interacting with from the nucleus have compensatory changes. That's selection. So I don't know. Always a troublemaker. Me, not you. Okay. So let's move on. Once again, all I'm doing here is giving you just a little bit of an overview of the richness of methodologies for testing, potentially testing for the alternative hypotheses. Okay? So how have people done that? But only a few ways. Please do see my book, you know, for other ways to do this that people have used. So this is one of the cooler ways. I really like it because it's a methodology actually I recommended recently to one of my professor, going to be much more well known than me, John Burke, who's working on lots of different kinds of plants. He's a professor actually at my university now, but he was a graduate student of mine. But this is something I recommended to John. So this figure is from Rasmo et al. Actually, obviously, but this is a paper about our species evolution and trying to test the alternative hypotheses that we have material from now extinct species of Homo in us, or there was ancient polymorphism, what they call population structure, and that's why we have these alleles shared between our genome and Neanderthalensis genome or Denisovans. Okay, so let me tell you why they're doing this. So in the initial, I may have mentioned this already in here, I'm sorry if I did, but in the initial two papers, the initial paper where they reported the Neanderthalensis genome, okay, from Svante Pabo's group, it was Green et al. And they also devised a methodology, it's the ABBA methodology, and the D statistic, okay? So if you go into that paper by Green et al. 2010, they give you the gory details in the supplementary section. And it's based around gene trees, locus by locus, looking for discordances, and it's based around us sharing alleles with Neanderthalensis that shouldn't be there. Okay, it's discordance. But it gives you a D statistic, and then it, or the D statistic gives you an estimate of whether or not you're looking at introgressive hybridization or not. Now, in addition to that, this was this paper in Science, a couple of papers, I think both of them were in Science, or one of them was in Nature, but in addition to that, they used some other things that we'll talk about in a moment. So they didn't just use the discordance. They also had other ways of inferring whether they were looking at introgressive hybridization. However, the D statistic has been shown, so that methodology, if you're using it, this is a caution to you. The D statistic has been shown to be very susceptible, affected by ancient or previous population substructuring, i.e. polymorphism. Okay? So keep that in mind. All right, if you're using this, a lot of people use it to test for introgressive hybridization, genetic exchange. All right, so what is this? So what we have here is if we had introgressive hybridization. What we have here is if we did not, but instead in Homo erectus, it gave rise to sapiens, and let's say, for example, if it did, I'm not sure, but Homo erectus gave rise to sapiens and Neanderthalensis. It's the ancestral species. If it had subdivision, i.e. it had some populations, lineages that had red chromosomes, i.e. alleles different, and then some populations, same species now, some populations had blue. Then if those admixed, it's still within the same species, okay? If those Homo erectus, let's assume it's that, admixed way back here, whatever, a couple of million years ago or whatever million years ago, then we're going to have some lineages by chance that have red and blue in them, okay? This is us, and this is us, and this is extinct, okay? So, and by chance also, you could end up with only red over here and only blue over here. This is incomplete lineage sorting, basically. Not basically, it's exactly coalescence, okay? So here, though, you have what looks like admixed chromosomes. Down here, you have ancient ancestry from here, not from introgression from Neanderthalensis here, right? So this is what you get. Now, the alternative hypothesis is that we did, and Neanderthalensis did, evolve from a common ancestor somewhere, a million years ago, 2 million years ago, whatever the new estimate is. But we diverged off and they diverged off. We had blue, they had red. And then about 50,000 years ago, we encountered Neanderthalensis and we had introgression. And then you had recombination, and then you end up, once again, some geographic regions of us do not have Neanderthalensis, or not much at all. This is Neanderthalensis. It's not around, but we sequence its genome. And then you have this, or the Homo sapiens populations. The difference here, this is junction analysis, the difference here between the size of those blocks on average, the mean size of those blocks and that, and this will be highly significantly, is theorized from the number of generations will be very small here relative to this, significantly different in size. Yes?
Also, like because there might be some degree of representation in the first place, whether it's introduced organization, would you see some parts which never get extincted?
Would we see?
Some parts which are always different in both populations and some which get
You mean between here and here? Or here and...
in the last case, we only have the log right that one, right? So some parts which are always like the
Yeah. Absolutely. You mean that like this, and it would be over here too. It's always, yeah, absolutely.
you wouldn't expect that from this person.
No, you would because you're going to. Well, I see what you're saying. I don't remember what they concluded about that. Whether the same empty areas, quote unquote, that don't have red, would they be the same? If these are all neutral, probably not. I guess I see what you're saying, but I don't know. I don't know about that. That's a good question. Would these ever, like that's always there in all populations? I assume it would be because recombination is going to be kept from breaking up certain gene combinations regardless of whether it's admixture or not. But, I mean, that's what we see in genomes in general. Yeah. But I see what you're talking about. That's a good question. If you modeled it out, would these move around, basically, and not be shared over here, ever one blue segment that's always there? Yeah. I don't know. I think so, but that's just intuition, and my intuition sucks. So, that's not based on knowledge. But what I want to say, too, about this, if you can do this, and like, for example, if you have pollen cores, we don't have these in our plants. If you're working on plants, or if you actually have some kind of remains that you can actually use and do this, it would be difficult and expensive. Okay, before I say this. But what they also have here that's so powerful to test this is that they actually have these intermediate samples of us, of Homo sapiens. And so they have a chronological, semi-chronological set of Homo sapiens fossils from here, close to this point, all the way to, of course, us, who are not fossils, I hope. And so they watch, the cool thing is they watch from the time point that they think that it should be introgressing down to here, they watch the blocks get smaller in those chronological. So I encourage some people who are working on certain kinds of plant species in China who did have pollen cores, or pollen samples from cores, that they think, is there a possibility of doing that, sequencing a sequential, because they're saying we have a homoploid hybrid species, or we have reticulate evolution, and that they have pollen cores from, you can't go way back, okay? You can't go millions of years. This is about the limitation. Anyway, so using junction analysis, or the block size analysis, this is another methodology. It's based around, initially, though, gene genealogy discordance. The only reason they're doing this is because they compared locus by locus Homo sapiens, or a plant species, with another species, and they got discordant gene genealogies. That's when you do this. Okay? Questions? Other questions, comments? Okay. All righty. One of the things that we can do, though, and it's one of the most powerful methodologies, I would argue, for this, and I'll refer back to the humans. I'll flip back there in just a moment. Let me just get this up here. Is using, geography. Okay, so once again, we're talking about, now these are eukaryotes, so it's introgressive hybridization or incomplete lineage sorting. Okay, and I'll give you another specific example, more recent, in a few minutes, that I've worked on. But what you have here in Africa, if you don't know, are a bunch of different kinds of baboons. They call them allotaxa. Some people give them a specific name, some subspecific. They're different behaviorally, morphologically, ecologically, et cetera. So whatever you want to call them, it doesn't really matter, and genomically. But you have these parapatric what are called parapatric overlaps. So they butt up against one another and they overlap in those regions. So the way that you can use geography in this case is you have these distributions like that. And if you look at the genomic constitution out here away from the zones, okay, in each one of these, and then look at the genomic constitution inside the zones, you can ask whether or not, it's what we always do to try to detect hybrid zones, is you ask geographically, are the overlap zones more heterozygous, basically? Okay, is there an indication more heterozygous than out here? Or is there more allele sharing in here of morphologies or whatever else you want to say in this region? And that may seem, well, yeah, it's a hybrid zone. Well, yeah, but that's using geography, okay? Geographic information. And I'm going to show you another one where we're using geography that's not this, quote unquote, simple per se. And that involves this. I'm sure you can't read this, but that's okay. Hopefully you can see the colors, okay? Seriously. So you have the yellows here, browns, greens, reds. Those are all samples of baboons, okay, from various places. And this is this new genus of primate, African primate, that was named, that was written up in Science in 2006. Now, I can guarantee you I would not get a paper in Science if I named a new species of iris, okay? And the reason they were able to do this is good reason, actually, I would say, and that is that, wow, this is amazing, a new genus of primate from Africa. That's sort of cool. And so I'm perfectly happy with them having it there. What happened, though, was that two now friends of mine, but then I didn't know who they were, Dietmar Zinner and Christian Roos, two scientists who were really started out as behavioral ecologists in working on African primates and Southeast primates, but Dietmar in particular works on baboons. And they had started drawing phylogenies from mitochondrial DNA, and as a lark, they used the DNA from Runguacebus, the sequence information that was out there on Runguacebus from just, there was only a couple of individuals who had had DNA collected from them. And they drew this tree. And lo and behold, and so I get this email and said, look, we don't know you, but we know you publish on natural hybridization and integration a lot, so would you help us think about these data? And I said, sure. So they sent this tree without the pretty colors, and they said, do you think that's a baboon, and that's Runguacebus. Do you think that's due to introgressive hybridization? I said, I have no clue. I said, it could be incomplete lineage sorting. What else do you have? What other information? And that's what they came up here, and they said, well, the reason we think it might be introgressive hybridization is because Runguacebus is paired, okay, with geographically adjacent baboons. It's overlapped with these areas. So it is like that parapatric area, except that there are so few individuals of this known, it doesn't look necessarily like a hybrid zone, okay? But it's probably just sampling error in one sense because Runguacebus has just not been extensively collected. So we argued that, yes, indeed, it was more likely than not that this was an introgressive hybridization event. At the same time, actually a sister paper, not from us, came out on this exact same data. Long story I won't go into. And they concluded, in contrast, that yes, this was hybridization, this reflected reticulate evolution, but they concluded that Runguacebus was a homoploid hybrid, which I don't buy. Okay, I really love having homoploid hybrids to report and discuss, and especially in animals, it's really cool to see how they evolved and all. But the reason I don't really buy it, and we said this in our paper, that we could have concluded that, but not really, is now in particular, they have a lot of this was based on mitochondrial DNA. Now they have nuclear loci, and this guy shows polymorphism, like you see in a hybrid zone where you have intergression. Some individuals are homozygous Runguicebus. Some individuals have Runguicebus mitochondrial DNA, but some alleles from another species, a baboon. Some individuals have baboon mitochondrial DNA and Runguicebus nucleus. It doesn't look like a homoploid hybrid that has a set genome/phenome, if that makes sense. But anyway, regardless, it's intergressive hybridization. Let me back up and tell you about here. I told you that also so they used this analysis, but I'll remind you that when Green et al. Published their paper and when, I can't remember who the lead author was on the Denisovans paper, but the Denisovans paper that also showed up in 2010, one of the things that they had to indicate, in addition to the discordance and the D statistic, was using geography. They said this apparently intergressed material is not in Africa, and it's not it only starts showing up in Europe, etc., where they predicted we would have first encountered Neanderthalensis. So our populations, geographically spaced out, are a mosaic of populations and tracts of these kinds of intergressed material. And so they use geography as part of their argument, part of their test for integration. Let's see. Sorry about this. Dink. Okay. And I think I've shown you this probably multiple times. Once again, remember, I'm just giving you a sort of a broad brushstroke in some ways, or very few examples, that's what I should say, of how people have tested it. So Myland et al. Used the Akaike information criteria. Do you use this at all? I know ecologists use it. I've never used it. But anyway, I've been told by my ecologist colleagues that, well, they didn't say it was stolen from us, but they just said, this is something that we commonly use. I've never used this kind of analysis. But basically, they model out, using this analysis, and the whole genome sequences of common chimp and us and Panpaniscus, or bonobos, okay? So they use the whole genome sequence, i.e. gene space. Remember, no whole genome has ever been assembled, okay? So gene space of these guys, and compared them. And they had a model with priors, etc., that said, okay, if you're less than 0, when you compare the various genomics regions overall, then what is the probability that you diverged in allopatry versus not allopatric? And this doesn't mean that they diverged in complete sympatric. It just means that they had episodic, probably, our proto-homo and proto-pon lineages occasionally intergressively hybridized. And so hopefully you can see everything below 0, it would agree with an allopatric model. Everything above, by their model, would be indicating divergence with genetic exchange for those guys. So one last, I think that's the last. Yep, that's it. So questions, comments about these? So this is just a few. I realize that, okay? Just a few methodologies that people use. There's analytical methodologies that I talk about in Chapter 3. Some of the very newer ones, at least they were new when I turned the book in to be, manuscript in to be printed. But they're no longer that new compared to what's come out. But anyway, questions or comments over this, and then we'll go back into just chapters 1 through 3. Okay. So chapter 1, history, concepts, what is all this about? Chapter 2 on species concepts and how that affects studies of genetic exchange, or can. And then also Chapter 3, once again, methodologies. So all of those are open for questions or comments. I have some other things I can talk about, but I really don't want to. I'd rather answer your questions. Yes.
Here that Anderson and Stebbins argued that natural hybridization likely caused the major chemistry reactions in the biological development. What was the
Say one more time.
examples were they talking about?
So that comes from, so the idea that, actually let's see. Yeah, in the rapid evolution talk that I gave you guys before I went to Pune. So what I mentioned this. So their paper was, and Stebbins and Anderson have argued this a bit. If you look at the biological record, there are bursts of, and this was their point, there are bursts of diversification, particularly after major mass extinctions. But there's also diversification that you see, there's also diversification that you actually see increasing at certain times, even outside of those mass extinctions. So when I teach undergraduate evolutionary biology, these patterns are quite striking. However, so what Anderson and Stebbins said was that those bursts, they're talking about the fossil record now, okay? They're really not talking so much about, they were trying to explain why could you get these kinds of bursts. Because if their basic point was countering the idea that, well, how do you end up with this kind of variation, these many lineages from bottlenecked units? So, which was going around during that 1950s because of Sewell Wright's work. Sewell Wright's work was, remember, genetic drift, genetic drift, genetic drift, and then they cross, and you get what Meyer talked about as a genetic revolution. That wasn't hybridization, though, to Ernst Mayr or Sewell Wright. Yeah, I need to reiterate that, but it looks very similar to it. So anyway, that's what they were saying, is that you needed. Stebbins referred to natural hybridization as just a special instance of mutation, right? And what he meant by that is, in our current terminology, would be that you take mutations in two divergent lineages that have added up, new mutations over however long. So you have a certain set of standing genetic variation in each one of them, and you put those together, and that's the mutation event. Yes?
So this is how to test the alternate hypothesis. So is it? It's sort of a crude method, but I think it might be useful. Mm-hmm. So for example, in your phylogeny, you have two distinct topology, and you have but two clade, which are two clade, which are together in one topology, but in other topology they are separate. And if you date these two trees, then you see that when they are separate, they have diverged quite a long time before, but when they're together, their separation is very recent. They diverged very recent.
Yes.
So based on this, can you tell that this is because of hybridization? I mean introgression, because they have separated quite a long time before, and very recently
Absolutely.
they have introgressed.
Yeah, very good point. That's absolutely what people do when they can do it. When they can date, they use, for better or for worse, they use a molecular clock kind of analysis, and they ask how many mutations, if this is a recent versus an ancient event. Actually, they normally, though, they're using multiple loci, so you're asking the mean, right? You're asking the mean age. And so in that case, yes. I mean, people do absolutely use that. They use that with our example of us. You know, they're asking what should the allelic divergence be if this is from ancient. Now they also use the block size, but from ancient versus recent. But you have to be careful with that. If I can get you to think, you know, all of us to think about this. Mm-hmm. So if you have ancestral polymorphism, okay, and that gets set, you know, put into Neanderthalensis and us as we're diverged. It's not sorting between them there, so they have different alleles. Mm-hmm. But those alleles are very ancient, right? And so when they come down and introgress, those alleles are the introgressed allele is going to look very ancient relative to the Homo sapiens allele. So that's how people actually do it. Does that make sense? So it's a relative, even if you don't have a time, it's a relative estimate of this allele in us. So you have to add this together. Let me back up. Let me show you what I mean real quick. Rather than doing hand puppets, which I don't do a good job of. Come on, be nice. So let's just use this. So what I mean by that is that relative to these alleles are very ancient, right? Potentially. Not necessarily. They could have arisen in this lineage, but so your introgression event can give you a signal of divergence here between your alleles. That's one outcome, right? But it also could give you a signal of both of these are ancient, right? Because they could have come in very similar times. So that's why we can't just date like one locus. We have to have a mean. We have to understand, this is my problem with molecular clocks coming out right now. Okay, I've never held a view that molecular clocks were very rigorous. So if you're using them, I understand we've done the same thing. Manuscript in review right now where we're trying to date when our species diverged, okay, and then putting big error bars around them. But, so dating these, well, this is stratified. You have those samples. But dating this event in anything always is going to have a gigantic error bar. Should. If it doesn't, you should worry. But there are other ways to do that. You know, are your plants separated in different places in the Himalayas? And you know that uplift was such and such. And you know these species have been over here and these species have been over here, but it looks like you've had intergressive hybridization. Well, maybe it's incomplete lineage sorting. Well, maybe it is a long distance migration. But there are other things, and then we know it's 10 million years ago or whatever. So we date it. We root it to that. So anyway, I don't know if that answers. Yes, people do it. That's exactly what people do quite often, is to try to figure out at these branches how long ago that was. I mean, we've been doing that since Vince Sarich and Alan Wilson produced that phylogeny that showed that we diverged from chimps and gorillas 4 to 6 million years ago, and they were actually correct. That was a molecular clock, but it wasn't based on DNA. Yes?
I'm a little confused by that because, like, what we just know, what we can see is just the red and the blue part, right? Which
Right.
is probably different kinds of. So how do we know that it is very much different? Because they could be very different and still in the same population as long as they're compatible with each other, right?
Well, what you're comparing it to, remember, are non-intergressed Neanderthalensis and non-intergressed us, if that's integration. And so what we're saying here is, yes, this could be very divergent here, but if this is very divergent compared to this or has a different pattern, okay, generally what we're really looking at is an allele that just doesn't seem to fit, because you have more than just the genomic material around that gene. You actually have a block. And that's the other thing you need to keep in mind. There's lots that goes into this. many pieces that are quite good because we can build up on this. So if you're looking, if you remember that paper that I showed you, I know these are way too uniform. So they're tree species, but this could be anything. But this is exactly what they've done here, okay? So you're walking down this piece of the genome. And as you walk down it, you're comparing, you're sliding your window down it, but you're comparing the genomes of two different things, two different plants, this populace or whatever, or Homo sapiens and Neanderthalensis. And what you actually generally see is not high divergence, but rather low. Both species turn out to be very similar here. But see, you're walking through here, okay, in this case, let's get down here. So you're walking through here, and these two, because of this integration, when you compare them, this is going to be very similar to that. So that's like that. And then when you get out of that and go to there, and you compare here, that's when you're getting these, right? So it's called outlier analysis. And so outlier analysis can also tell you why we may be different from Neanderthalensis in morphology or physiology or biochemistry, because if you get a peak like that's outside, way outside of the mean, then we use this to tell us why we're different from chimps. They're doing this right now. Okay, what loci really look like they were under directional selection. But this would indicate generally is what we use. But what I was saying was we have to be careful because when we compare this with this or this with this, maybe the alleles are just similar. Once again, it's an incomplete lineage sorting versus integration. If, let's say these were both real similar to one another, then the question would be, how long ago might the event have been? Was it up there where we got both of that red and blue? Because part of it will be, right? Part of this is going to be due to incomplete lineage sorting. Nobody's arguing, well, they shouldn't. Nobody's arguing that all of this is integration. But on top of this, remember they've been looking at the geographic setting, geographic spatial outlay of integressive hybridization in our populations or populace or anything else and saying, does it make sense? Are they overlapping? Would they have overlapped there? And then on top of that, because we have genomic information, we know what genes are in here. Why would it have integressed, potentially? Is it neutral? Once again, remember, my estimates from what I've seen in my own plants, just to take them as an example, say 40% of what gets across acts like it's neutral. Okay? 40% or something like that or more is stopped. It doesn't integress. And then the rest looks like it's adaptive. It's usually smaller than that amount. But so if that makes sense, so we're having to test all of this. We have the ability now to look across all this. And I personally think that we should. Many people choose not to because they want to simplify it, and I understand, but okay, other questions? Yes. Could. Mm-hmm. Absolutely. So this was something that, you know what? This is going to lead into, he's asked about Klein theory. Could we go ahead and take our break? This is something I actually wanted to do. It's a little early. This is going to be a longer little presentation I do about hybrid zone theory and Klein theory. So if we could do that after the break, that would probably be better.
That's fine. We can stop right now.
Okay.